Bom dia. Bom dia a todos. O Instituto Nacional de Traumatologia e Ortopedia tem a honra de recebê-los para o primeiro encontro do Ciclo Internacional de Atualização e Infecção Ortopédica. Sejam muito bem-vindos. Esse é um evento que visa promover a difusão de conhecimento científico atualizado, o intercâmbio entre diferentes áreas do conhecimento e a reflexão crítica sobre práticas contemporâneas no manejo das infecções osteoarticulares, contribuindo para o fortalecimento da assistência, da formação profissional e da pesquisa aplicada. Em nome da comissão organizadora, gostaria de fazer um agradecimento especial à FAPERJ, à Sociedade de Infectologia do Estado do Rio de Janeiro, CIERJ, Sociedade Brasileira de Ortopedia e Traumatologia Regional Rio de Janeiro, equipes do INTO, ARCOM, Auditório, DIEMP, ARPES, AINF e DITRO, a Margarete Catoia, que ajudou a gente com consultoria em relação ao Headcap, e a todos os mais de 600 inscritos ao evento. O formato do evento será de 3 palestras em torno de 20 minutos cada, totalizando 1 hora, então de 7h30 às 8h30, seguido de 30 minutos de discussão. Por isso, anotem suas perguntas e a gente fala no final, tá bem? Para darmos início ao nosso ciclo, gostaria de apresentar a mesa de honra dessa solenidade de abertura. Dr. José Paulo Gabi Aramburu Filho, diretor do INTO. Dr. Marcelo Serrão, presidente da SBOT Rio, que falará online. Doutora Carla Regina Oliveira de Moura Ronchini, presidente da Sociedade de Infectologia do Estado do Rio de Janeiro, também falará online. Dr. Cláudio Feitosa de Albuquerque Júnior, chefe da Divisão de Traumatologia e Ortopedia do INTO. Doutora Juliana Arruda de Matos, infectologista do INTO e presidente da comissão organizadora do evento. Agora passo a palavra para o nosso diretor e dele a palavra segue para os demais integrantes da mesa.
Bom dia a todos. É uma honra estar aqui participando, um prazer ver um evento tão concorrido, mais de 600 pessoas inscritas. Eu estava falando com a Juliana que o presidente da Sociedade Brasileira de Cirurgia do Joelho, assim como o Marcelo Serrão, que está online, que é o presidente da Sociedade de Ortopedia do Rio de Janeiro, ambos nos procuraram, pediram, mostraram interesse em participar do evento, quer dizer, dando total apoio ao evento e dando a importância que realmente o evento tem para a gente. Todos que estão aqui, eu acho que muitos sabem que na minha posse eu coloquei como uma das metas o tratamento, a prevenção, enfim, uma atenção toda especial ao tema infecção. Então, eu quero parabenizar enormemente a Juliana, a você, Kelly, e a todos os envolvidos da AINF, enfim, da Sociedade de Infectologia, que proporcionaram esse evento de alto nível técnico, com um ciclo de palestras, não é um evento só, e que eu tenho certeza que vai de encontro ao que o INTO se propõe em termos de educação continuada, em termos de desenvolvimento, de tecnologia, principalmente em relação ao foco nosso na gestão, que é dar uma atenção toda especial à infecção. Parabéns a vocês.
Bom dia aí, saudando a presença de todos presentes, e online. A importância que a infecção ortopédica tem, está sendo dada agora, essa mudança de chave. Eu, há 30 anos, cheguei aqui no INTO como R1, e acompanhei o hospital esse tempo todo, e a gente finalmente dando a importância que o tema infecção tem no mundo e no nosso hospital, que impacta tanto a nossa vida profissional e a vida dos nossos pacientes. Então, muita felicidade essa mudança de chave aí, essa importância dada à infecção, que a ortopedia agora abriu os olhos para esse tema tão importante. Bom dia.
Então, bom dia a todos e a todas. É um prazer a gente estar aqui nessa manhã, fazendo esse evento que a gente preparou com muito carinho para falar desse tema que é tão importante, que angustia todos nós que lidamos com os pacientes, que lidam com esses problemas de infecção osteoarticular. Desde o começo, quando nós abrimos as inscrições, foi uma grande procura, uma grande surpresa, mais de 600 inscritos, demonstrando como que esse tema é importante, como ele é relevante. Existem muitas lacunas de conhecimento nesse assunto, então a gente precisa realmente discutir, debater, como o Cláudio falou, a gente superar aí um certo tabu, porque é um assunto que só a gente discutindo, estudando, conhecendo as evidências, reconhecendo as lacunas e procurando suprir essas lacunas, que a gente vai conseguir, de fato, melhorar a qualidade de vida, prevenir essas infecções, melhorar o diagnóstico, o tratamento e a recuperação dos nossos pacientes. Esse evento foi pensado com muito carinho, pensando em privilegiar a questão do acesso, né? Então, a gente pensou nesse formato, em várias aulas curtas, ao longo do ano, num horário cedo, depois que você já deixou a criança na escola, antes de entrar no centro cirúrgico, disponível online, em formato híbrido, depois disponível no YouTube, para quem não conseguiu assistir de forma síncrona, com tradução na linguagem brasileira de sinais, justamente para a gente poder aumentar essa acessibilidade, para que todo esse conhecimento possa ser espalhado e atingir o maior número de pessoas possível. Tá? Então, eu queria agradecer, em nome da comissão organizadora e científica do evento, a presença de todos e todas.
Então, a gente vai começar o evento e depois eles se pronunciam, os presidentes da CIERJ e da SBOT Rio de Janeiro. Agradecemos às autoridades presentes, desfazemos a mesa para o início das palestras. Eu chamo para iniciar o Ciclo Internacional de Atualização em Infecção Ortopédica a doutora Juliana Arruda de Matos. Ela é médica infectologista do INTO, pesquisadora em saúde pública do Instituto Nacional de Infectologia Evandro Chagas Fiocruz, doutora em doenças infecciosas e parasitárias pela Universidade Federal do Rio de Janeiro, membro da Sociedade Europeia de Infecção Osteoarticular e delegada do Consenso Internacional de Infecção Musculoesquelética desde 2018. A palavra com Juliana. Informamos que a presença será contabilizada de duas formas. Para os que estão prestigiando presencialmente, será através do QR Code, que já apareceu e vai aparecer outras vezes na tela durante o evento de hoje. Para os que estão online, de forma síncrona, pelo link que vai ser fixado no chat do YouTube. Atenção, os links vão ser desativados, né? Um pouquinho depois do término do evento, isso será repetido em cada dia de evento ao longo do ano, e a certificação de participação será dada apenas aos que estiverem presentes de forma síncrona, presencial ou virtualmente, a pelo menos 4 eventos mensais até dezembro, tá?
Bom, então eu vou falar hoje para vocês um pouquinho sobre os critérios diagnósticos de infecção articular periprotética, o critério do Consenso Internacional de 2018, ou da Sociedade Europeia de Infecção Articular, e a nova proposta de consenso unificado que está sendo posta pelas diferentes sociedades. Bom, então, o contexto. As artroplastias são procedimentos custo-efetivos e com elevado impacto na qualidade de vida dos pacientes com doença articular avançada. No entanto, mesmo os implantes modernos têm uma vida útil limitada, levando à necessidade de procedimentos de revisão dessas próteses, né? A gente sabe que a revisão pode acontecer por diversos motivos, e as revisões que são por causas infecciosas, por infecção, têm um pior prognóstico e exigem um tratamento específico, diferente das causas não infecciosas. No entanto, nós não temos um exame padrão ouro que possa definir de forma precisa entre causas infecciosas e causas não infecciosas. Em alguns casos, essa diferenciação é muito óbvia, como a gente pode ver nos dois casos iniciais, mas em alguns casos isso não é tão fácil de se definir se há uma soltura asséptica ou uma soltura séptica. Bom, então, visando resolver esse problema, foram propostos, a partir de 2011, critérios definidores de infecção de prótese articular por diferentes entidades. O primeiro, mais difundido, mais utilizado, foi aquele publicado em 2011 pela Sociedade de Infecção Musculoesquelética. Em seguida, em 2013, esses critérios foram revisados na primeira reunião do Consenso Internacional de Infecção Musculoesquelética, em 2013. Nesse mesmo ano, a Sociedade Americana de Doenças Infecciosas também lançou os seus critérios, são 2 critérios diferentes, concomitantes no mesmo ano. Em 2018, aconteceu a segunda reunião do Consenso Internacional e os critérios foram novamente revisados. E, em 2021, a Sociedade Europeia de Infecção Articular lança os seus critérios esses que foram endossados por algumas outras entidades, como a própria Sociedade de Infecção Musculoesquelética e o grupo de estudos em infecções associadas a implantes da Sociedade Europeia de Doenças Infecciosas e Microbiologia. Eu vou falar, então, sobre esses dois últimos critérios, que são os que estão mais usados no momento, né? Nos trabalhos científicos e tudo mais. Então, vamos lá. Eles têm algumas diferenças bem importantes. Tá? Vamos falar primeiro dos critérios do Consenso Internacional de 2018. Essa é uma tabela traduzida, resumindo o consenso. Os critérios maiores são a presença de 2 culturas positivas com o mesmo micro-organismo fenotipicamente, gênero, espécie e antibiograma, ou a presença de fístula comunicando com a articulação ou visualização direta da prótese. Qualquer um desses 2 critérios isoladamente confirma a infecção. Na ausência dos critérios maiores, a gente se dedica a olhar critérios menores, baseados em marcadores inflamatórios séricos, como a PCR ou o dedímero, ou ainda a velocidade de hemossedimentação, marcadores inflamatórios no líquido sinovial, então a contagem total de leucócitos, ou a alfa-defensina, que é um marcador, um biomarcador, ou a presença da esterase leucocitária, que nada mais é do que a fitinha de IAS, então aquela bolsinha que marca a presença de leucócitos na urina, né, na fitinha de IAS, pode ser utilizado você pingando o líquido articular ali e vendo se aparecem 2 cruzes marcando a presença de leucócitos no líquido articular. Tá? Ou ainda o percentual de polimorfonucleares. Observem que para esses marcadores, esses biomarcadores inflamatórios, existem pontos de corte para infecção aguda e crônica nesses critérios. A gente pode ainda lançar mão dos achados do histopatológico, então, se houver mais de 5 neutrófilos em 5 ou mais campos de grande aumento, na membrana periprotética, é importante atentar para o espécime clínico, a membrana periprotética, isso também é um critério menor de diagnóstico de infecção por esse consenso. O achado de purulência intraoperatória ou uma única amostra de cultura positiva dentre as diversas que devem ser colhidas para o diagnóstico desse tipo de infecção. Então, cada um desses critérios você atribui uma pontuação e o somatório dessa pontuação vai dizer se o paciente está infectado, se for 6 ou mais pontos, não infectado 0 a 2 pontos ou inconclusivo entre 3 e 5 pontos. Importante lembrar, é importante não confundir os critérios do Conselho Internacional com esses critérios desse artigo que foi publicado um pouco antes, que serviu de base para o consenso, mas não é o critério do consenso, tá? Parece, mas não é. Então, tem algumas diferenças importantes, né? Essa tabela ela divide entre diagnóstico pré-operatório e intraoperatório e não divide entre aguda e crônica, porque esse artigo ele foi feito apenas com pacientes com infecção crônica, tá? Excluindo, então, as infecções precoces e as hematogênicas agudas. Tá? Então, não vamos confundir um critério com o outro. Essa é a tabela traduzida do consenso da Sociedade Europeia de Infecção Osteoarticular. Vocês vão observar que tem diferenças importantes. Ele divide os critérios em 3 grandes categorias. Categoria A, os critérios clínicos, que incluem achados clínicos e o achado de proteína C reativa. Critérios de laboratório, que incluem os achados em líquido sinovial, microbiologia e histologia. E o critério C, que é de imagem, né, levando em consideração achados de medicina nuclear. Tá? Os pacientes podem ser classificados, de acordo com esses critérios, em infecção improvável, quando todos os achados são negativos, infecção provável, quando você tem 2 achados positivos, sendo um do critério clínico e outro do critério laboratorial, ou um do clínico e um do critério de imagem, ou pode ser infecção confirmada, quando você tem um achado de qualquer um do critério A ou B dessa coluna em vermelho. Tá? Mantive aqui como critério de infecção confirmada. Nós temos os mesmos critérios do consenso, então fístula ou visualização da prótese, ou 2 amostras positivas para o mesmo micro-organismo, também são critérios maiores, porém, esse sistema de critério da Sociedade Europeia considera outros critérios também como critérios que sozinhos já definem e confirmam a infecção, como, por exemplo, o achado de mais de 3.000 leucócitos no líquido sinovial para a Sociedade Europeia, sozinho, isoladamente, já configura um critério. Né? tem outros achados que também geram esse diagnóstico. A infecção provável, você deve levar em consideração critérios clínicos, como, por exemplo, uma história de afrouxamento precoce da prótese com menos de 5 anos de instalação, ou uma história de problemas de cicatrização da ferida, associado a algum achado laboratorial, usando pontos de cortes mais baixos, né, que são pontos de corte menos específicos, que têm maior sensibilidade. Então, você junta um aspecto clínico com um aspecto laboratorial com menor especificidade, maior sensibilidade, e você junta dois aspectos e você gera um diagnóstico de infecção provável. Você não tem tanta certeza assim, mas você tem duas evidências com menor especificidade que juntas te dão esse diagnóstico. Ou ainda você ter um achado clínico com uma cintilografia de leucócitos marcado positivo, também gera esse diagnóstico de infecção provável. Quando você tem tudo negativo, a infecção é considerada improvável. Tá? Então, resumindo a diferença, no critério de 2018, nós temos provas inflamatórias com diferentes pontos de corte para infecção aguda e crônica, enquanto no EBGIS você não diferencia agudo e crônico. O que você diferencia são pontos de corte com maior sensibilidade para infecção provável, então maior sensibilidade e menor especificidade, e pontos de corte com maior especificidade, mais rigorosos, que quando presente você tem maior especificidade, você confirma a infecção, tá certo? Lembrando, mais uma vez, que esses pontos de corte para infecção aguda do consenso, eles não foram validados, tá certo? Não foram validados naquele estudo que serviu de base para essa definição da pontuação. Outra diferença importante, esterase leucocitária, dedímero e VHS só é considerado no consenso internacional, enquanto que história clínica pregressa, critérios radiológicos e medicina nuclear são considerados apenas no EBGIS. E com relação ao diagnóstico microbiológico, o consenso internacional já sinalizava, não dentro do critério propriamente, mas numa observação que, em caso de cultura negativa, métodos de sequenciamento de nova geração poderiam ser utilizados. Isso não é mencionado no critério do EBGIS. Por outro lado, o EBGIS leva em consideração a sonicação da prótese explantada e a visualização de micro-organismos no histopatológico. Tá? E aí a gente se pergunta, se tem tantas diferenças, qual que é o desempenho comparativo desses critérios? Existem 2 artigos que comparam diretamente os 2 critérios. Tá? Então, esses 2 artigos eles comparam diretamente os critérios. Né? E o que os 2 artigos concluem que o critério da Sociedade Europeia ele é mais sensível do que o do Consenso Internacional. Isso é uma coisa que a gente já intuía pelo fato de ter mais critérios confirmatórios do que o consenso. Né? Ele também gera uma quantidade menor de diagnóstico inconclusivo, né? Que diagnóstico aquela coluna do meio. Então, isso dá mais certeza diagnóstica para a equipe. E ele também tem um melhor desempenho pré-operatório. Tá? São conclusões muito parecidas que foram que esse outro estudo chegou. Tá? Então, também o menor número de infecções intermediárias pelo EBGIS do que pelo Consenso Internacional, uma definição mais sensível para o EBGIS e mais precisa no pré-operatório. E uma coisa importante que esse artigo mostra, ele fez uma comparação muito interessante. Ele seguiu os pacientes durante 2 anos e procurou ver se o EBGIS é mais sensível, que ele faz mais diagnóstico de infecção do que os outros critérios, será que esses pacientes realmente têm infecção ou será que eles estão fazendo um diagnóstico a mais, são falso positivo? E o que esse estudo mostra é que os pacientes que foram diagnosticados com infecção pelo EBGIS, mas não foram diagnosticados com infecção pelos outros critérios, de fato evoluíram pior, mostrando que esse diagnóstico realmente é preciso. Tá? Mas a gente continua com um problema se a gente tem 2 critérios aceitos de forma simultânea. Né? A gente tem diferentes padrões na pesquisa e confusão entre os médicos, as pessoas que assistem os pacientes. Então, as sociedades se reuniram para chegar a uma definição unificada de infecção de prótese. Então, as diferentes entidades, Sociedade de Infecção Musculoesquelética, a Sociedade das Infecções dos Estados Unidos, a Europeia de Infecção Articular, o Consenso Internacional e o Grupo de Estudos de Infecção e Implante da Sociedade Europeia de Doenças Infecciosas. E aí, depois de muitos trabalhos, né, muitas revisões sistemáticas e várias rodadas de critério Delphi, eles chegaram às seguintes conclusões. Esses critérios não foram publicados ainda, eu sei que eles já estão em vias de ser publicados, todas as sociedades já deram o seu endosso, tá? Então a gente deve ver esses critérios sendo publicados em breve. Ele é dividido em 2 conjuntos de critérios. Os critérios independentes, que são aqueles que isoladamente fazem o diagnóstico de infecção, e é muito parecido com os critérios maiores do EBGIS. Tá? Então, a fístula comunicando a articulação com o meio externo, que se desenvolve ou persiste após a incisão ter cicatrizado ou quando já deveria ter cicatrizado. Né? Achados de microbiologia: 2 culturas de tecido periprotético positivas com o mesmo micro-organismo, ou uma cultura positiva de líquido sinovial ou fluido de sonicação, mais uma cultura positiva do tecido periprotético. Então, aqui a sonicação que entra sempre junto com um material periprotético e não como critério isolado. Essa é uma diferença para o EBGIS, tá certo? Marcadores inflamatórios: leucócito acima de 3.000, polimorfo nuclear acima de 75, ou uma histopatologia positiva. Tudo isso na ausência de uma explicação alternativa. Esses critérios todos apresentam especificidade acima de 95%, portanto, a presença de apenas um critério desses de forma isolada já faz o diagnóstico de infecção confirmada. Já um outro conjunto de critérios, que eles chamam de critérios de suporte, é onde residem as principais diferenças desse critério unificado. Então, achados de microbiologia: uma única amostra positiva de líquido sinovial, fluido de sonicação ou tecido periprotético. Isso já era antigamente, né? Mas a novidade: um teste molecular positivo para qualquer micro-organismo em líquido sinovial, tecido ou fluido de sonicação. Então, entrando aqui o resultado de teste molecular. Exames de imagem: a cintilografia com leucócitos marcados positiva, também já estava presente antes. Novidade: o PET-CT positivo quando realizado mais de 6 meses após a artroplastia índice, também entrando aí como um critério de suporte. Marcadores inflamatórios: contagem de leucócito entre 1.500 e 2.999, polimorfo nuclear entre 65% e 74%, ou qualquer biomarcador sinovial alternativo positivo. E aí vem outra mudança importante, porque é considerado, não sei se vai dar para ler, está um pouquinho pequeno, mas eu vou ler para vocês. A esterase leucocitária positiva, né, 2 cruzes, proteína C reativa no líquido sinovial, que nunca tinha entrado em critério, passou a entrar. A alfa-defensina, já estava presente antes. A calprotectina, que também não existia antes. Levando em consideração que esses pontos de corte podem variar de acordo com as orientações do fabricante. Tá? Então, qualquer um desses biomarcadores serve como um critério de suporte. São os critérios que têm especificidade acima de 80%. Então, a gente vai considerar uma infecção como provável na presença de um critério microbiológico mais um critério laboratorial ou de imagem. Tá? Esse conjunto de critérios também prevê fluxogramas de diagnóstico para auxiliar no fluxo de exames e de investigação para 3 situações. Quando você tem a infecção precoce pós-operatória, eles propõem um fluxo diagnóstico. Para as infecções mais tardias, é um outro fluxo, levando em consideração as diferenças e especificidades de cada tipo de infecção. E um terceiro fluxograma, que é para as infecções agudas hematogênicas. Bom, gente, eu acho que eu devo já ter passado um pouquinho do meu tempo, então eu não vou ter tempo de entrar em detalhes nesse fluxo, a gente pode discutir isso melhor na discussão. Eu agradeço muito a presença de todos, peço desculpas pelas intercorrências aí. Gostaria de lembrar que nós teremos o Congresso de Controle de Infecção brasileiro aqui no Rio de Janeiro em novembro, gostaria de convidar a todos. E agradecer muito a presença de todos e todas. Obrigada.
Robin Patel. Ela é uma profissional com ampla trajetória na área de microbiologia. Ao longo da sua carreira, já publicou mais de 540 artigos, com destaque para diagnóstico de doenças infecciosas, especialmente infecções ortopédicas, tema ao qual se dedica há décadas. A doutora Patel é microbiologista, ocupa o cargo de professora Elizabeth Robert Allen de Medicina Individualizada, além de atuar como professora de microbiologia e de medicina na Clínica Mayo. Atualmente exerce a função de vice-presidente de Educação no Departamento de Medicina Laboratorial e Patologia da instituição, além de dirigir o Laboratório de Pesquisa em Doenças Infecciosas. Nesse contexto, desenvolve estudos voltados a biofilmes, resistência antimicrobiana, infecções articulares periprotéticas e métodos diagnósticos bacterianos. Assistiremos à aula dela gravada para evitar problemas de conexão, mas a doutora Patel volta e estará conosco depois para a discussão, tá bom?
Thank you so much for having me here today. I'm excited to talk about the microbiological diagnosis of periprosthetic joint infection, including molecular and metagenomic methods in clinical practice. I've been working in this area for many, many years, and hopefully I can share some of what I've learned about this and some of where the field is in terms of the diagnosis of periprosthetic joint infection. I visualize periprosthetic joint infection as a biofilm-mediated infection where microorganisms adhere to the surface of the implants and grow in biofilms, which are phenotypically different than planktonic microorganisms. The organisms grow slowly. They elaborate an extracellular polymeric matrix. They're also very resistant to many of the antibiotics that we use in our host immune system as well. But I think this is really important in terms of diagnostics. So when I visualize periprosthetic joint infection, again, I think about organisms contacting the surface of an implant, such as illustrated in this cartoon, and adhering to that surface and growing in this biofilm state. And I think this is important for diagnostics because it really helps inform where we're going to go looking for the organisms if we want to find them in periprosthetic joint infection. I also think about how this might impact diagnostics in terms of how these biofilms might trigger the host immune system to respond to them and how we can use that host immune system response to develop diagnostics to know that those organisms are there on the surface of the implant. And so a lot of what we understand about the host immune response, I think, is very basic today, but we know that there are a lot of neutrophils, for example, present at the site of periprosthetic joint infection, and I'll incorporate that as I talk about diagnostics. I think as a microbiologist, it's also very important to understand which organisms we're looking for when we go looking for them. It's almost like you go hunting, you got to know what you're hunting for so you can use the right tools to find what you're looking for and also to understand what it is that you've detected. The microbiology of periprosthetic joint infection can be very diverse, but there is sort of a standardized list that we can look at that represents the most common causes of periprosthetic joint infection. And that includes mainly aerobic gram-positive bacteria, including mainly the staphylococci, both Staphylococcus aureus, as well as the coagulase-negative staphylococci, especially Staphylococcus lipinensis and Staphylococcus epidermidis, which are particularly common, but also streptococci, especially Streptococcus agalactiae, enterococci, especially Enterococcus faecalis, and Corynebacterium species, especially Corynebacterium striatum, but others as well. Conversely, at least in our practice, aerobic gram-negative bacteria are somewhat less common. I realize that may be somewhat different for you, that you may see more aerobic gram-negative bacteria, so you need to keep that in mind. And that includes both members of the Enterobacterales, such as Escherichia coli or Klebsiella pneumoniae, and Pseudomonas aeruginosa. Anaerobes are not uncommon as well, and so this includes organisms like Cutibacterium acnes and Feingoldea magna, and that's important because if we're trying to grow up these organisms in culture, we need to use anaerobic cultures to find anaerobic bacteria. And then at the very bottom of this list are fungi and mycobacteria, and you can see they're much less common. I'm going to come back to that in terms of diagnostics and tell you a little bit of a story about that later on. Now, we sometimes think about these infections as involving one microorganism, but in fact, many of them, up to a third, can be polymicrobial. And when we look at polymicrobial periprosthetic joint infection affecting hips and knees, some of the common organisms that we see present with others are the non-aureus staphylococci, enterococci, corynebacterium species, and Feingoldea magna. Now, I've been working in this area since the 1990s, and our team, as well as others, have helped to advance the diagnosis of periprosthetic joint infection. And this is just a graphic of the numbers of papers published on the diagnosis of periprosthetic joint infection over time. And you can see that they've really risen up. So that's good, right? We have a lot more data. We much better understand how to diagnose this infection. And the first point that I think is really important is that we need to consider periprosthetic joint infection as a disease unto itself, a disease that needs its own diagnostics, separate from the diagnostics that we might use for native joint septic arthritis or non-foreign body associated osteomyelitis. I think what we've learned is that if we apply diagnostics that are used for other diseases to periprosthetic joint infection, they don't work well. Now, as much as we'd love to have good blood tests that detect periprosthetic joint infection, mostly this is a localized infection, so most of our diagnostics will focus on specimens collected from around the site of an implant. So that will include synovial fluid, periprosthetic tissue, as well as the implant itself, and I'll be focusing on some of those. I always think there are two questions we want to answer when we're thinking about diagnosis of periprosthetic joint infection, two big questions. There might be some smaller questions as well. The first question is this joint infected? Yes or no? Because you can have arthroplasty failure for non-infectious reasons, and those cases obviously don't need antibiotics, and they don't need to be managed as if you're managing an infected case from a surgical perspective. That differentiation is sometimes clear, as you know, but sometimes it's a little bit more ambiguous. Once we believe that an arthroplasty site is infected, then it's important to define the infecting microorganisms. And why is that? That is because it tells us which antibiotics we would use to treat that particular infection. And perhaps in some cases, it could help us direct surgical strategy as well. So those are the big questions that I think about answering. And we can talk about, once you know the organism, you also need to know its susceptibility and get into some more subtle pieces. But those are the big questions. And we can look at either host response or we can look at detection of microorganisms to try to answer those questions. From the standpoint of trying to determine, is this infected or is this not infected, synovial fluid is a great first step to start with because it's relatively easily collected, certainly more so than periprosthetic tissue or the implant itself. And synovial fluid analysis can be done using a multiplicity of different tests. But the easiest test to do is a cell count, looking at a total nucleated cell count, as well as a neutrophil percentage. These are old-fashioned tests that are available in almost any lab. What's important is that the cut points for differentiating periprosthetic joint infection from non-infectious arthroplasty failure are different than the cut points for native joint septic arthritis. I'm excited to tell you that sometime in the near future, there will be new criteria for diagnosis of periprosthetic joint infection called the Unified PJI definition that will be coming out in a major medical journal. Unfortunately, it's just a little bit too early, and I can't tell you exactly what those are or where they will be, but look for them coming out soon. You can also look at the American Academy of Orthopedic Surgeons guidelines on the prevention and diagnosis of periprosthetic joint infection as another area to look for interpretive cutoffs for total nucleated cell count. But this is a pretty good biomarker for periprosthetic joint infection, as is the neutrophil percentage. And I'm showing you the cut points on the right-hand side and the unified PJI definition for diagnosis of periprosthetic joint infection. Of course, we can argue about exactly where that cut point should be, but that's what you're going to see in that new definition. Now, obviously, there are a number of other tests you can run on synovial fluid. One of the tests that I get asked about a lot is alpha-defensin. Alpha-defensin is an antimicrobial peptide that is made by neutrophils and other cells, and it has been shown to be a very good biomarker of periprosthetic joint infection in synovial fluid. And in fact, in the United States, there's an FDA-cleared assay, which is configured as a lateral flow assay that looks a lot like a COVID test, for running on synovial fluid to diagnose periprosthetic joint infection. I think this is great, and we have a number of studies that look at the performance of alpha-defensin, and it's very good. But in many ways, its performance reflects the performance that you would get from cell count and neutrophil percentage. So it's not adding a lot in terms of a host response biomarker. I'm going to talk a little bit later on about some more advanced host response biomarkers. But I know you wanted me to talk about molecular diagnostics, and as you know, that's one of my passions is developing and offering molecular diagnostics. So I want to turn to what we can do with synovial fluid, since we're talking about synovial fluid here. So this is the 16S ribosomal RNA gene. It's present in all bacteria, and bacteria are by far and away the most common cause of periprosthetic joint infection. This is also present in mycobacteria, so it takes care of the rare mycobacterial infection as well. And so you can design PCR primers to amplify and then sequence this target. And that can be done on synovial fluid. So that's pretty exciting. And you might think, well, that should be the way that we diagnose periprosthetic joint infection. Well, I've been working in diagnosis of periprosthetic joint infection for some time. And I will say we can certainly do this from a methodology standpoint, but the findings are a little bit disappointing. This is a study that we published in MBio in 2022, and we took synovial fluids from patients with hip, knee, shoulder, and elbow arthroplasty failure. There were 154, 36 had periprosthetic joint infection, and we compared 16S ribosomal RNA gene PCR and sequencing to culture. So there are two things that I walk away with looking at this. First of all, you always need to do synovial fluid culture. That's very important. It is not perfectly sensitive. In our study, it was about 72%. And that's why people want to turn to molecular diagnostics, because we have culture negative periprosthetic joint infection. But look at what happened with synovial fluid 16S ribosomal RNA gene PCR and sequencing. The sensitivity was no better than culture. If you do them together, you boost up your sensitivity a little bit, but you still have those microbiologically negative cases. So, it works. You can do it. Is it perfect? No, unfortunately. So don't worry. We have other tests that we can do. Now, recently in the United States, at least we have had FDA authorization of the BioFire joint infection panel. So this is a multiplex PCR panel that is cleared by the United States Food and Drug Administration to test synovial fluid. These are the organisms that are on it. So they include a number of bacteria, Candida species, and Candida albicans, as well as resistance genes. This is really not specifically for periprosthetic joint infection diagnosis. It's for testing synovial fluid. And so we asked the question, how would it perform in cases of periprosthetic joint infection? And we looked at 60 synovial fluids from patients with knee arthroplasty failure, and we compared this panel to 16S ribosomal RNA gene PCR and sequencing and culture, classifying our cases using IDSA diagnostic criteria. And what we found is that for organisms that are on the BioFire panel, it performs very well. So the positive percent agreement with culture was 91% for the BioFire panel, and it was 96% for 16S ribosomal RNA gene PCR and sequencing. But the panel did not do well across the board. So the overall positive percent agreement was only 56% in this small study. And that's because there are organisms that are causes of periprosthetic joint infection that are not on this panel. And a classic example that I highlighted in red here is Staphylococcus epidermidis. So the panel doesn't have Staphylococcus epidermidis on it. If your patient has Staphylococcus epidermidis periprosthetic joint infection, it's going to return a negative result. And that's a false negative result really for the patient, although technically the panel is not detecting it. So it's just really a gap in the panel, not necessarily a gap in the performance of multiplex PCR. So maybe we can use better panels, right, in the future. But still, I mentioned that synovial fluid is not perfectly sensitive in terms of advanced diagnostics, not just culture, but 16S ribosomal RNA gene PCR and sequencing. So many, many years ago, we really thought about how can we come up with an ideal specimen in which to detect the organisms that are causing periprosthetic joint infection. And I will say, I don't think this is a perfect specimen, but we were really thinking about how can we go where the organisms are? And as I mentioned in my little cartoon at the beginning, that's on the surface of the implant. So if surgeons are removing implant components, we can take those into the lab and we can sample biofilms on the surface of those implants. And so we devised a method for sonicating implants. And what I'm showing you here is what we do in our lab clinically every day to diagnose periprosthetic joint infection. So the surgeon puts the implant components in a sterile jar, sends them to us in the laboratory. We add a salt solution, we vortex that, we sonicate them in a bath sonicator. Pretty simple machine, the same kind of machine you would use, say, to clean jewelry or blinds or electronics. And that takes some of the biofilms off the surface of the implant and also disaggregates them. And then we vortex and we do centrifugation step and then semi-quantitative plating. And we did a large study of hip and knee arthroplasty failures that we published in the New England Journal of Medicine many years ago in 2007. We looked at a total of 331 patients resected implants. And we showed that sonication of those implants was more sensitive than sampling periprosthetic tissue for culture for diagnosis of periprosthetic joint infection and equally specific. And we've done similar studies as cited here with shoulder arthroplasties, elbow arthroplasties, spine implants, and there have been a number of other studies that have been done as well. So this is another way of trying to find the organisms that cause periprosthetic joint infection, but there are a couple of caveats about it. First of all, if you're using implant sonication, you do need to use some sort of cutoffs in terms of what you're calling positive. You cannot consider any positive, like a single colony that grows up from implant sonication, to represent a positive result, because if you do that, you're going to have some false positive results because there can easily be a colony of bacteria that shows up in your culture. So we use semi-quantitative reporting. I'm not suggesting you have to use exactly what we use, but what we use is shown here. And what is shown is that if you use semi-quantitative reporting, your specificity is very high. Now, you might look at this and say, well, wait a second, that sensitivity is not great. And that's a challenge in periprosthetic joint infection. So we continue to work, and I'm going to show you some of the data. I'm trying to find those organisms that are causing periprosthetic joint infection. So we'll come back to that. Now, the other specimen type, besides synovial fluid and the implants themselves, is periprosthetic tissue. That's tissues from around the site of the implant. And surgeons can collect that at the time of surgery, and then those can be analyzed for bacteria. As I mentioned at the beginning, because anaerobes can cause periprosthetic joint infection, we have to do aerobic as well as anaerobic cultures. And we know that some of those anaerobes are slow growing, so we need prolonged incubation. Also, historically, we were told based on guidelines that we needed 5 or 6 tissue specimens from every patient. I'll come back to that with some newer information there. But all of that means a lot of work for the microbiology laboratory. And so that over time, we're seeing more and more cases of periprosthetic joint infection due to the number of joint replacement surgeries being performed. And so we tried to look at whether we could make our culture process more efficient in the laboratory by culturing tissues in blood culture bottles. And we did a big study on this. We enrolled 369 subjects who are undergoing hip, knee, shoulder, or elbow arthroplasty revision. About a third of them had periprosthetic joint infection. And we simply homogenized their tissues and did our standard plate and broth cultures that's shown on the left-hand side, but also took an aliquot of that tissue homogenate and put it into an aerobic and anaerobic blood culture bottle. And then we compared the results we got. And our hope was that the results would be similar. And then we could switch to doing our cultures in blood culture bottles, which is much easier for the laboratory because the bottles just sit on the instrument. And if there's growth in them, they signal positive. And if not, they just go through their incubation period and at the end get pulled off and reported as being negative. We didn't find what we were looking for. We actually found something better. The cultures in blood culture bottles were more sensitive than the regular plate and broth cultures. Not what we were looking for, but a pleasant surprise from this particular study. They also did decrease laboratory staff time. So this is what we were really after. They were more efficient to do our cultures this way, which was pretty exciting. And then lastly, we were able to take the data from this study and look at how many tissue cultures you need to perform from each patient to establish a diagnosis of periprosthetic joint infection. And what we did is we took the data from that large study and then we randomly down selected around the number of tissue samples. And we showed that if you're using cultures in blood culture bottles, you need 3 tissue specimens, not 5 or 6, to establish a diagnosis of periprosthetic joint infection. And if you're using conventional plate and broth cultures, which some labs do, and that's fine, you need 4. You don't need 5 or 6. So this is, I think, pretty exciting because now we better understand how to use our tissue cultures in clinical practice. But having improved the way that we do tissue cultures, we went back and did another study comparing tissue cultures in blood culture bottles versus sonication of the resected implants. And what we showed is that there was still an incremental increase in sensitivity with the sonicate fluid cultures versus the tissue cultures, but the best sensitivity was combining both of those together. And so if you really are looking hard for the cause of periprosthetic joint infection, doing both tissue cultures, and if you can do sonicate cultures or sample the implant surface in some other way, this is what would be recommended. Now, I want to take a step back here and tell you this story about fungal and mycobacterial stains and cultures because I think it is pretty exciting in terms of what was found in this study, and it also makes a really nice story. So there was a fellow, an infectious disease fellow, Gino Tai, who came to me when he was here at Mayo, and he said, I really want to do this study on fungal and mycobacterial stains and cultures in periprosthetic joint infection. And I said to him, well, you don't need to do that because they shouldn't be done. We already know that answer. And he said, I really want to do the study and see what I find. And so he went ahead and did the study, and what he found was really interesting. So he looked at hip and knee periprosthetic joint infection from 2010 to 2019. There were 2,067 periprosthetic joint infection episodes. And in our clinical practice here at Mayo Clinic, there were 3,629 fungal and 2,923 mycobacterial cultures done, which was astounding to me. So clinicians like to order fungal and mycobacterial cultures, and at least one of them was done in 56% of episodes. So this was amazing because I didn't think they should be done in the first place. But then he looked at the test positivity rate for fungal and mycobacterial cultures. Those were 5 and 1% respectively. But remember, that's not saying they are the cause of periprosthetic joint infection. That's just a positive result. Overall, there were 40 true fungal and 8 true mycobacterial periprosthetic joint infections. But here is what was really surprising to me. I just described how we do our tissue cultures in blood culture bottles. That process, the tissue culture and blood culture bottles, was 90% sensitive for diagnosing true fungal periprosthetic joint infection and 100% sensitive for detecting rapidly growing mycobacteria. So you don't really need specialized fungal or mycobacterial cultures to find these organisms, which are rare. They can show up in your regular bacterial cultures if you do them correctly. Also, he looked at fungal stains. They were done in 27 true fungal periprosthetic joint infections. 4 were positive for a 15% sensitivity. That's very, very low. And no mycobacterial stains were positive. Now, I want to talk about some advanced sequencing-based diagnostic work that we've done looking at sonicate fluid. So I mentioned that for me, this is the specimen type in which we have the highest likelihood of finding an organism, but we still don't have very good sensitivity. So we first designed our own PCR panel for testing sonicate fluid. This was based on the most common causes of periprosthetic joint infection, and we showed that boosted sensitivity a little bit to 77% versus 73% with culture, but we still have microbiologically negative cases. So here we turned to the technique of shotgun metagenomic sequencing. So this is a technique that is very exciting for clinicians because you can detect anything. You take your sample, you sequence everything in the sample, and then you look at what you find. So when you sequence everything in the sample, and we're just looking at DNA here, we're not looking at RNA because we don't think RNA viruses are really relevant to what we're studying here. But what was found was, what you can find with shotgun metagenomic sequencing is obviously human DNA, bacterial DNA, fungal DNA, even parasitic and viral DNA. We don't really expect to find those, but that can be found as well. You can find everything very agnostically. And so we executed what is kind of the largest or largest study of shotgun metagenomic sequencing. We looked at 408 sonicate fluids from patients with arthroplasty failure, and we compared results to culture. So you see 3 rows here. The first row is the non-infected patients, and you see mostly we're not detecting anything by shotgun metagenomic sequencing. The second row is the culture positive cases, and you see that we're mostly detecting the same thing we were finding in culture. The third row is what's most interesting. So those are our culture negative periprosthetic joint infections. So again, we're doing shotgun metagenomic sequencing on sonicate fluid, and we were able to find something in 44% of cases. These are the organisms that we found. So just to walk you through this, in the first row, I'm showing the organisms that were found by culture causing periprosthetic joint infection that were not picked up by shotgun metagenomic sequencing. So it's not perfect, right? The second row is organisms we found in the non-infected cases, which is interesting. The third row is additional new organisms that we found in the culture positive periprosthetic joint infections, suggesting that some of them are maybe more polymicrobial than we think. And then the fourth row is the organisms that we found by shotgun metagenomic sequencing in the culture negative periprosthetic joint infections. And those, by and large, look like classic causes of periprosthetic joint infection with some exceptions. So this is one of the cases of 408 that we looked at. I just want to show it to you to show you an example of what maybe this can do. This is a 53-year-old man who had a right knee periprosthetic joint infection. He was aspirated multiple times. Always his synovial fluid looked infected based on cell count and differential, but it never grew anything. And so he came to us for a 2-stage resection arthroplasty. We, again, looked at his synovial fluid. The results are shown there. It looks infected, but the cultures were all negative. He did undergo implant sonication. He also received 6 weeks of intravenous cefepime and vancomycin and had a vancomycin and gentamicin spacer placed. We were doing our research study at that time, so what you see on the bottom is what we found by shotgun metagenomic sequencing in his sonicate fluid. You see that we saw a lot of reads, 27 million reads, most of which are human. But if we take those out and we look at the microbial reads, most are bacterial. And if we take the bacterial reads, most of them belong to an organism called metamycoplasma or mycoplasma salivarium. At the time, that organism hadn't been reported as a cause of periprosthetic joint infection. It does not grow well in culture. This was a research study. So he underwent reimplantation of his joint. We were generating this data on the side. And unfortunately, he failed that reimplantation. And when he came back, we did some specialized cultures and recovered metamycoplasma salivarium in culture. So for these really unusual causes of periprosthetic joint infection, certainly a molecular sequencing-based approach can be very helpful. But back to the beginning where I talked about 16S ribosomal RNA gene sequencing versus what I just shared, shotgun metagenomic sequencing, these are 2 different strategies. The shotgun metagenomic sequencing approach, which is shown on the left, is much more complicated and expensive to execute. The targeted metagenomic sequencing approach, for example, just looking at the 16S ribosomal RNA gene, which is present in all bacteria, which are the main causes of periprosthetic joint infection, is still complicated, but a lot simpler than shotgun metagenomic sequencing approach. And so we then asked the question, if we took our sonicate fluids and we ran them both ways, shotgun metagenomic sequencing and targeted sequencing, would one be better than the other? And I think this is really helpful. So we took a subset of almost all the sonicate fluids from the shotgun metagenomic sequencing study that I just shared, and we ran 16S ribosomal RNA gene PCR sequencing. And we basically showed that the positive % agreement of the 2 was the same. And so I don't think you need to do shotgun metagenomic sequencing in clinical practice. We did that as a research study, trying to answer some more basic science questions. But I think if you really need to go generically after any bacteria, using the 16S ribosomal RNA gene can be the way to go. Certainly less expensive than a shotgun metagenomic sequencing-based approach. So I'm going to wrap up here. I have several messages to walk away with. First of all, diagnostics for periprosthetic joint infection need to be specific for periprosthetic joint infection. We can't just use generic diagnostics for this disease, and that applies to culture as well as culture-independent diagnostics. Hopefully you appreciate that periprosthetic joint infection is a biofilm-associated infection, and the implication from a diagnostic standpoint is the closer we can get to the surface of that arthroplasty, including taking the arthroplasty itself, the more likely we are to find microorganisms. I also shared that molecular and metagenomic approaches can be helpful. I think importantly, we still need to do culture. We can't just evolve to using molecular diagnostics, but I also showed you that these molecular diagnostic approaches are not always perfect. So we still need, better understanding, better development of these approaches. And I'd like to thank the huge team of investigators and the patients who participated in this, and the NIH and CDC for funding my research lab. Thank you very much.
Eu vou passar a palavra, aproveitar enquanto eles colocam a Carla e a Patel aqui, para dar a palavra ao Marcelo Serrão, que é o presidente da Regional do Rio da SBOT, para falar.
Bom dia a todos. Primeiro, queria pedir desculpa pelo atraso aí, me atrasei uns 10 minutos. A gente tem consenso em relação à infecção, mas não tem consenso em relação ao trânsito do Rio de Janeiro, né? E eu acabei me atrasando um pouquinho. Parabenizar o Into, ao Gabi, toda a equipe do Into pelo evento. A infecção ortopédica, a infecção e artroplastia, principalmente, é um assunto muito importante, muito impactante, não só para a saúde pública e privada, como também para o cirurgião, né? Porque é um drama para o cirurgião ortopédico ter um caso de infecção e pode ter um desfecho dramático para o paciente também. Então, essa iniciativa de vocês é uma iniciativa fantástica. Eu espero que continue aí durante muito tempo. A gente tem muita coisa para aprender em relação à infecção. Você vê que o último consenso do ano passado. E, por último, eu gostaria de convidar a todos para o nosso evento de artroplastia, Congresso Internacional de Artroplastia, que será esse ano no Windsor Barra, nos dias 31 de julho e 1º de agosto, e a gente vai ter um módulo só de infecção, só para tratar desse assunto. Muito obrigado a todos e bom dia.
Gente, a Carla teve que entrar, a presidente da Sociedade de Infectologia do Rio de Janeiro teve que entrar numa aula, então ela não vai poder falar, mas a gente quer abrir para discussão, aproveitando a doutora Patel, e eu vou refazer a mesa com Osama e Juliana para as perguntas. A gente tem pergunta no YouTube. Alguém quer perguntar aqui do auditório? Alguém quer fazer alguma pergunta? Não fiquem tímidos, estamos em casa.
Eu vou responder uma pergunta que apareceu no chat. Gente, primeiro...
A pergunta foi se falso positivo seria possível para o diagnóstico de infecção periprotética aguda pela cintilografia de leucócitos marcados.
Então, essa é uma grande limitação dos testes de medicina nuclear de um modo geral, né? Porque eles têm dificuldade de diferenciar entre a resposta inflamatória pós-operatória e a resposta inflamatória provocada pela infecção. A metodologia com leucócito marcado é a que tem melhor especificidade.
A Juliana perguntou para ela sobre os diferentes métodos moleculares, né, qual a diferença entre eles, e você acha que isso é só válido para culturas negativas ou não?
Ela falou das vantagens e desvantagens, de dificuldade de sensibilidade do BioFire, por exemplo, que os métodos eles não substituem as culturas, que eles não são também essa panaceia, essa solução para todos os problemas, né? Mas então, aonde que a gente usa exatamente? É quando a cultura é negativa? É quando você precisa de uma resposta rápida? É enquanto a cultura não fica pronta? Quando que a gente usa na prática? A gente escolhe fazer esses métodos, inclusive considerando que são métodos caros, né? Então, esse detalhe da aplicabilidade prática, eu queria ouvir da professora Robin. So, nice. So we are here also. Robin, thank you so much for accepting our invitation.
Oh, can you hear me? Oh, I didn't realize. Sorry, I'm texting you now.
But so I see there's a question that says, what is the role of different molecular methods? Would you say only for negative culture infections? So this is a really good question. I mean, I think we all think molecular methods are the answer to everything. But as I highlighted in my presentation, they're not perfect. They're not universally available. And really, we need to turn to cultures as our first diagnostic. We should always do cultures. And if cultures are positive and are quite convincing, for example, if you have 2 positive cultures for the same microorganism, you've really arrived at defining the microbiologic cause of PJI. And I don't see why you need to perform a molecular diagnostic on top of that. If you would not have a concern that you found the wrong organism, I suppose you could say, well, what if there's something else there, which, could be possible. But ordinarily, if you have the diagnosis, I think you're finished and you don't need to turn to doing additional molecular diagnostics. Now, conversely, if your cultures are not positive and you have a case that you believe is infected for reasons other than culture, obviously, then that could be a case where you would turn to molecular diagnostics. And then I guess the question was, what is the role of the different molecular methods? I highlighted in my presentation that we have several different molecular methods. Of course, we have different specimen types, synovial fluid, tissues from around the implant, sonicate fluid if you are sonicating an implant. And then we have PCR diagnostics. We have targeted sequencing, for example, of the 16S ribosomal RNA gene. There are other also broad-range genes in bacteria, or metagenomic sequencing. I'm not aware that metagenomic sequencing is necessarily broadly available, so mostly people are talking about PCR, such as a PCR panel, or a targeted sequencing type of approach. The nice thing about the PCR in the US, we have the BioFire panel for testing synovial fluid. The nice thing about that is it's easy to use and it's fast. The limitation, as I highlighted, is that it doesn't include all the organisms that cause periprosthetic joint infection. So it's great if your case happens to be caused by an organism on the panel, but if not, then it's not terribly helpful. The specificity is very high, though. And so a lot of people will then turn to a 16S ribosomal RNA gene PCR sequencing type of approach if the cause of PJI is not known. So cultures are the first diagnostic that should be performed to diagnose PJI. And I think that's good, right? Because that's the test that most laboratories have for this diagnosis.
Tá bom. Agora, o Osama fez uma outra pergunta. Você quer falar sobre isso?
Eu perguntei para ela sobre um problema que a gente tem no quadril, que é pré-operatoriamente fazer uma aspiração e essa aspiração vir seca, né? Sem líquido sinovial. No joelho não é um problema tão grande assim, mas no quadril a gente acaba enfrentando bastante esse problema. Eu perguntei para ela se ela tem alguma dica de como proceder nesses casos, né? Como é que aumenta a sensibilidade disso, como é que aumenta a quantidade de líquido coletado, e nos casos de punção seca, qual é o próximo passo que ela orienta a gente tomar, né?
Okay, so the next question is, we have problems collecting synovial fluid at hip replacements. Do you have any tip to increase the amount of fluid collected? And in cases of dry aspiration, what is the next step you use? So obviously this is a challenge, right? If you can't get the specimen that you would like to get to analyze, to culture, and to assess cell count and differential, then that's a problem. According to the literature, one approach that could be helpful is to use imaging-based guidance that could help you overcome a difficult aspiration, but clearly hips are more difficult to aspirate than knees. And it could be that in the end you just, are unable to obtain synovial fluid. In that case, there's not much that you can do preoperatively. Of course, you can obtain synovial fluid intraoperatively, and I would say that is probably your choice there. So you're performing a surgery and you can collect not only tissues from surrounding the implant, but synovial fluid at the time of surgery. It doesn't help you with preoperative planning, but it's still synovial fluid, so it's a way around that.
Vou falar um pouquinho aqui da nossa rotina, né? Como é que a gente procede aqui essas coletas no serviço. A gente aqui coleta 5 amostras de tecido periprotético, né? De diferentes locais, ou seja, não adianta pegar o tecido do mesmo local e dividir em 5 frascos, né? E a gente acaba não conseguindo inocular tecido nesses frascos de hemocultura. A gente inocula líquido que aparece no local, hematoma, líquido sinovial, né? E a gente envia nesses frascos de hemocultura, mas realmente tecido a gente tem muita dificuldade de inocular nesse frasco. Primeiro porque os tecidos são difíceis de coletar com uma agulha, né? Então o orifício do frasco é muito pequeno para essa inoculação. Então a gente acaba enviando os tecidos em frascos convencionais e o líquido que a gente envia nos frascos de hemocultura. E sempre a gente prima por coletar membrana periprotética ou algum tecido ósseo.
Tem algumas perguntas aqui no chat do YouTube, estão perguntando muito a questão que ela falou da coleta de 3 espécimes só, né? E a questão de inocular em frascos de hemocultura, estão perguntando como é que ela, exatamente esse protocolo dela, e que espécimes são esses que ela coleta, de que locais ali no momento cirúrgico que ela coleta. Acho que dá para compilar algumas perguntas falam sobre isso.
Okay, yes. So I think the question is about tissue cultures and how many tissues to collect for culture. And I'll just go over the history here and why we collect multiple cultures and what we know about this. So this really stems back for many, many years when people started to try to diagnose periprosthetic joint infection, and they realized that tissue cultures were not very sensitive to diagnose periprosthetic joint infection, but also that you could have a positive result that was a false positive result due to an isolation of an organism that could be a contaminant, such as Staphylococcus epidermidis or a Cranibacterium species, but could also, in the case of periprosthetic joint infection, be a pathogen. And so you need the multiple tissues for 2 reasons: to maximize sensitivity as well as to address that question of specificity. I always say do not send a single tissue culture to the laboratory. That's going to just create confusion because of the low sensitivity and also because of the specificity problem. But then the question you're asking is about how many specimens to send to the laboratory. So originally, if you look back on the IDSA diagnostic guidelines, which are very old now, these said that you needed 5 or 6 tissue specimens from around the implant. And certainly that's possible that you could collect that. In fact, we see sometimes when our surgeons collect even more than that. You may see that occasionally in your laboratory. But really the question is, what is the right number of specimens? And we must keep in mind that methods to do tissue cultures have improved over time. Maybe some of those improvements are small, maybe some of them are larger, but some of the sensitivity challenges have been improved. For example, we know that we need to routinely be doing aerobic and anaerobic cultures, and then we also know that for anaerobic cultures, and to a little bit of an extent for aerobic cultures, we need to prolong the duration of incubation, maybe a little bit more than you would use for native joint septic arthritis. I'm not saying that for aerobic cultures you would do, say, a 14-day incubation, but maybe a few days of incubation as opposed to just an overnight incubation. So in the lab, we've improved a lot of things with cultures. And then I described in my presentation how in our lab, we culture our tissues in blood culture bottles. And the reason that we do that primarily is because it's just easier for us in the laboratory. We put the tissues into the blood culture bottles and then the instrument monitors them. And we don't need to pull them out and take a look at the plates and broths every day for several days. It's semi-automated in a way, but it also does help us improve our sensitivity. So we looked back on how many tissues you need to establish a diagnosis of periprosthetic joint infection, and we showed that if you're doing plate and broth cultures, you need 4. And if you're doing the cultures in blood culture bottles, you need 3. Now, that's what we advise. I think it's also important to note that we would also advise synovial fluid cultures be done in every case. It makes sense. And of course, even if you have an organism in a synovial fluid culture, you should still do tissue cultures because, you know, according to guidelines, and I'll get to that in just a minute, you ideally want 2 positive cultures for the same organism. So you can't get that with a single synovial fluid. Of course, you could re-aspirate the synovial fluid and approach it that way. But mostly you want to go to those tissue cultures. And you can factor in sonication cultures in that equation if you're using sonication cultures. I have good news for you, Juliana. The paper was accepted in the New England Journal of Medicine between the time I recorded the lecture last week and now. So what I want to share with you is that there will be new diagnostic guidelines for periprosthetic joint infection that will be published soon. I don't know when, in the New England Journal of Medicine. Juliana, I think you covered that in your lecture earlier, or will cover that. And so it's great news. This was an international group that put together those guidelines. I will say I think they're great because the guidance was put together in a very rigorous way. But in many cases, we really didn't have the data to perfectly answer the questions that we were asking in the way that we wanted to answer them. And so I'm going to digress just for a minute here. You know, it's a publication on the diagnosis of periprosthetic joint infection. And ideally, if you're asking a question, for example, about culture, you classify your cases without culture, and then you ask how culture performs. But culture is such an intricate part of the diagnosis of periprosthetic joint infection that it's actually hard to find that kind of data, and it's actually hard to do. And so we don't have perfect data in this space. Of course, we never have. That's been my journey working with periprosthetic joint infection. But I think it's the best data that we have today. And it was also put together by a group of people from around the world, including people working in the laboratory, infectious diseases physicians, and of course, orthopedic surgeons. So it's a very multidisciplinary group as well. And it's endorsed by a number of societies, as you'll see. And I think Juliana talked about that. Juliana, I saw there was also a question that was added that says, how do you put tissue inside the bottle? This sounds like putting genie inside a bottle or something like that. Well, when we do tissue cultures in the lab, even when we do plate and broth cultures of tissues coming from any site, including native joints or prosthetic joints, we have to homogenize them first before we put them on plates and into broth. And that can be done in a number of ways, actually. So you can use homogenizers. So that's what we do in our laboratory. You can use a mortar and pestle. You can use bead beating. There are a number of ways to, if you will, mince up or grind up tissue or homogenize tissue. And then you just take that sort of slurry and pick it up and inoculate it into a blood culture bottle.
Great news. Gente, então recebemos aqui informação em primeiríssima mão. O artigo do Consenso Unificado acabou de ser aprovado na New England. Foi aprovado da semana passada para hoje. Então teremos em breve o artigo publicado lá. Mais alguma coisa, gente? Acho que não. Gente, então acho que a gente pode encerrar. Eu queria agradecer mais uma vez a presença de todos e todas aqui no auditório, também online. O nosso próximo encontro, que está marcado para o dia 6 de maio, vai ser um encontro sobre prevenção, inclusive por ocasião da Semana de Higiene das Mãos, que a gente comemora no dia 5 de maio. Então a gente vai trazer o tema tão importante, tão relevante, que é a prevenção da infecção de prótese articular, né? O que funciona, o que a gente não tem certeza ainda que funciona, quais são os princípios por detrás da fisiopatogenia dessas infecções que vão nos guiar no que a gente vai fazer para poder prevenir essas infecções. Então, a gente está preparando a programação com muito carinho. Teremos os detalhes disponíveis em breve. Muito obrigado, bom dia a todos.